In this video, we are going to look at the regulation of gene expression in eukaryotes. In the previous video, we saw how prokaryotic gene expression is regulated through operons, and we saw how we have repressible and inducible operons present in prokaryotes. In eukaryotes, there are multiple layers of regulation, and we've discussed quite a few of them as we were discussing transcription and translation. And so let's go through this, and some of this is probably going to be a revisit. So DNA has, as we saw, there's it's very, very long sequences of DNA within every cell, and yet they are placed in a very, very compact space inside the nucleus. That's because of the way that DNA is organized, into these really tightly packed structures. So the key to this is the presence of proteins called histones, which have a lot of positively charged amino acids. And DNA is an acid, as the name indicates, right? Deoxyribonucleic acid. So it's an acid, and as you know, acid and base, positive and negative charge, can interact with each other. So the DNA is wound around the histones, and in the form of almost like strings on a bead. And this is the structure that is present for most of the cell cycle. And this is called as, those structures are called as nucleosomes, right? When you have DNA wound around histones. And then the nucleosomes are then wound around each other in the form of coils. So this is a structure which we call as chromatin fiber. So this is what it is. What is this? The stage it is found in through most of the cell cycle. The structure, the chromatin fibers, can be folded back when they have to condense into these looped domains, and forming these 30 nanometer fiber loops, attaching to a scaffold which is made of a different set of proteins called non-histone scaffolding proteins. And then finally, those loop domains, 30 nanometer fibers, are condensed back on each other like this to form chromosomes in the during the stage of mitosis, just before prophase starts, or during prophase you have condensation happening. So why are we discussing this in the context of gene regulation? Because when your DNA is compacted in the form of chromosomes, the transcription machinery, the RNA polymerases will and the transcription factors will not have access to it. But when it is loose, as in these cases, then there is more access for the transcription machinery. So physically, the compaction of DNA prevents it from being transcribed. So that is one of the ways that gene regulation happens in eukaryotes. So there's a name for that. So when you have chromatin that is highly condensed, which is, you can actually see this with the light microscope. That is when it stops the chromosome from being, from the DNA from being accessed by the RNA polymerase. Therefore, transcription cannot happen. Whereas, and that condensed form is called as heterochromatin. So we need to remember that. Heterochromatin is a condensed form of DNA. Euchromatin is the less compact form of DNA, where your transcription factors can actually access your DNA, and therefore transcription can happen. And that is a good way for cells to regulate which part of the chromosome is going to be transcribed or not transcribed. So if a particular part of the DNA needs to be transcribed, then the DNA moves from heterochromatin stage to euchromatin stage, and that allows the RNA polymerases to access the DNA. But how does the cell decondense a DNA from a heterochromatin stage to a chromatin stage? So that happens because of either modifying the histones by adding groups as acetyl groups to them. So acetyl group is the CH2CH3. So you can add acetyl groups to them, to the histones, and when you add acetyl groups, it goes from tightly packed to less tightly packed, and so histone acetylation causes the DNA to be more easily transcribed. When you remove the acetyl group, and that's called deacetylation, then the DNA is not transcribed because it becomes more condensed. So that is one of the ways that it is regulated, by acetylation. By adding an acetyl group, it becomes less condensed. When you remove the acetyl group, it becomes more condensed, and this process is called as acetylation and deacetylation. The other way is to change the DNA itself. So you know there are 4 nucleotides, right? A, T, G, and C. By changing the C, so the cytosine, by adding a methyl group to it, it causes more condensation. So basically, the whenever you add a whole bunch of methyl groups to the cytosines, it takes a heterochromatin state, which means the DNA is not accessible to RNA polymerases. Now, the interesting thing is this methylation events are inherited, meaning these lifetime experiences, constant exposure to something, constant stress or whatever, can cause certain genes to be not expressed because of methylation. And even though this is not a change in the actual sequence of the DNA, but what we are changing is the ability of the DNA to express a gene or not express a gene. You are actually suppressing a gene or overexpressing a gene just by changing the accessibility of the RNA polymerase by methylation. And that can be inherited. So this is called as epigenetics. So epigenetics is changing the inheritable characteristics or traits of an organism by changing the accessibility of RNA polymerase to it. So without changing the DNA sequence. So this is all stuff that we have looked before, the types of RNA and what transcription is, what translation is, what is a coding strand and what is a non-coding strand. So we have looked at all of that. Now, take a look at the gene one more time. So you have your gene over here, and that includes the promoter region as well. Remember, promoter is a place where your transcription factors can bind, and along with other proteins can recruit the RNA polymerase and therefore cause the transcription to happen. There are 2 types of elements when it comes to regulation of gene expression. One are the proximal elements, like the promoter, which is present. Proximal means close. So proximal elements which regulate the expression of genes, because binding of the transcription factors to the promoter can influence transcription, and promoters are found very close to the gene. They are called as proximal elements. And then you have the distal elements. Distal means far, and these are sequences of DNA that are far removed, far away from the gene itself, but as we'll see in a minute, they can influence how transcription happens. So they are called distant elements. And distal elements are usually enhancer elements. So we'll talk about them in a second. So we know the process of transcription, how it happens. As we saw, proximal element control elements are close to the promoter, and distal control elements may be far away from the gene and located within an intron. So enhancers are groupings of distal control elements. Can be more than one distal control elements, but they are present far away from the gene. And there are proteins called activators. We saw how transcription factors bind to promoters and cause RNA polymerase, recruit RNA polymerase to make messenger RNA. There are proteins called activators that bind to the enhancer region. Again, remember, enhancer regions are distal elements, a cluster of distal elements that can influence transcription of a particular gene. And activators are proteins that can bind to an enhancer region and And what happens, and we'll see this in a picture, what happens is the gene, even though there's a lot of DNA in between this region where the gene is and where the enhancer regions are, what it's going to do is it's going to bend over by elements called as DNA bending elements, and that's going to bend and then come back and bind to the transcription factors and enhance the activation. Or in fact, what it can do is, by the bending of the DNA and the activator coming close to the promoter region, it can actually help assemble the transcription factors at the promoter region, at the data box, therefore enhancing transcription. So those are activators. Activators are elements that bind to enhancers, and enhancers are collection of distal elements that influence transcription. And you also have repressors. Repressors, we saw what repressors are in prokaryotic cells. Same thing here, you have repressors that can inhibit the expression of a particular gene. And sometimes these activators and repressors can change the chromatin structure, make it from euchromatin to heterochromatin, which is slightly wound to heavily wound, or from heterochromatin to euchromatin. So they can have those indirect effects on regulating gene transcription as well. So here's an image. We see enhancer far away from the gene. You have your gene, you have your promoter sequence, and you have your distal control elements. A collection of these is the enhancer, and your activator comes and binds to it. Then a DNA bending protein comes, it bends, and it causes the assembly of mediator proteins and allows the binding of general transcription factors, which recruits the RNA polymerase II, which causes synthesis. Let's see if we can see a picture image of this.
The central dogma of molecular biology: DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
So in case you couldn't get the audio, what it was showing was initially the assembly of a transcription initiation complex, and that included the activator regions or activator proteins attached to enhancer regions coming down, and then helping the mediator proteins get the RNA polymerase in place so that transcription can happen. So transcription happens. We already saw how transcription happens. Another way of regulation of gene expression is splicing and capping and addition of poly A tails. So again, we have seen all this before. So splicing and alternate splicing can lead to variations in what genes are which of the messenger RNAs are going to be translated and in what form. So that again is a way of regulation of how much of proteins are produced. We saw this before, alternate splicing. In this example, they have shown where there are totally 5 exons within this gene, but of those, one variation of this has only 1, 2, 4, and 5 exons, and this variation has 1, 2, 3, and 5 exons. So that's how you're able to get a whole variety of proteins from a limited number of genes. We also looked at the codon chart previously, and what the start codons are and what the stop codons are. So now we have RNA being made into proteins through translation, and again, we have seen how all of this happens. Translation, I don't want to repeat that again, and how termination happens. So there is one more way that translational regulation happens. One is before translation happens, the messenger RNA that is produced after splicing, mature messenger RNA, in whatever form that is, it can be the product of alternate splicing, however that is. So the messenger RNA that is produced can be regulated. And one of the ways that happens is called RNA interference. RNA interference is mediated by a group of small RNA molecules called microRNAs. And microRNAs are usually about 21 nucleotides long, very, very small. And when the microRNAs they're made from a larger pre-microRNA sequence, and then they are cleaved or diced into small microRNA sequences, which is about 21 nucleotides long, or 21, 24, 23, depends. So those nucleotides, those short microRNA sequences, are bound to proteins to create a microRNA protein complex, and this complex can bind to complementary messenger RNAs. So if I have a messenger RNA that needs to be regulated, then the levels, if you have a whole bunch of messenger RNAs being created, and if there are microRNAs that have complementary sequences to the messenger RNAs, then they can bind to the complementary sequence, meaning that the messenger RNA is no longer available to bind to the translational complex, protein translation complex, or binding to this complex can also cause the messenger RNA to be cut into tiny, tiny pieces, because that's what this protein complex will do. So because of complementarity between the microRNA and the messenger RNA that is present naturally, you can create you can create one more strategy for regulating the levels of messenger RNA there. And finally, we have the post-translational gene control method. So protein is made now. What next? How do you control the amount of protein there? So one of the ways this happens is through the ubiquitin proteasome complex. So ubiquitin is a protein that is present everywhere. Okay, so and it acts like a tag. When it sees a protein that needs to be degraded, ubiquitin will go and bind to the protein to be degraded. So this ubiquitinated protein is now sent to a protein structure called as a proteasome. And a proteasome is like a protein machinery. It takes in the ubiquitinated protein and cleaves the protein into tiny peptides. And the peptides are not wasted. They are broken down into amino acids, and they are used for making other proteins. So that is one more way of regulation. So I'll just quickly go over the different strategies. First thing we saw for eukaryotic gene regulation is how you have histone proteins binding to DNA to make nucleosomes and then chromatin fibers, and you have the open form of DNA, which is Euchromatin, and you have the closed form, condensed form, which is heterochromatin. By regulating how much of heterochromatin is there, or which sequence of the DNA of your chromosome is going to be heterochromatin or euchromatin, you can allow access to RNA polymerase. If it's heterochromatin, RNA polymerase cannot bind. If it is euchromatin, RNA polymerase can bind. The addition of acetyl groups can cause heterochromatin formation. Removal of acetyl groups, which is deacetylation, can lead to opening up of the DNA. Methylation, which is change in the cytosine nucleotides of methylation of cytosine nucleotides, can cause condensation, basically forming of heterochromatin. And if you remove the methyl groups, demethylation, then it becomes more open. So the methylation processes and histoneacetylation can be carried over across generations, from one generation to next generation, even though there is no change in the DNA sequence. So this regulation of gene expression, that some genes are expressed and some genes are not expressed in individuals because of life experiences without any change in the DNA sequence, is called as epigenetics. And that is an increasingly important field in biology. And then we saw the other way of DNA regulation is using distal and proximal elements. Proximal elements are those that are close to the gene, and that includes transcription factors that bind to promoters and causing the expression of genes. The others are enhancer sequences, which are collection of distal elements that can bind to proteins called activator proteins, and activator proteins can come and bind to the can help with the assembly of the transcription initiation complex and cause RNA polymerase to move forward. You also have repressors in eukaryotic systems that block the transcription process. And then we also saw the post-transcriptional, or I'm sorry, post-transcriptional modifications. We saw we have splicing, and we also saw where we have capping, and we have poly-A tail additions. And then pre-translational modification are when you have the messenger RNA, the mature messenger RNA binding to binding to the microRNA, and therefore preventing the formation of the translational process or degradation of the messenger RNA. That is one. The other way is is post-translational modification. So after the protein is made, and if the protein is not functional, or there's a mistake here or whatever, then it has to be degraded. First, it binds to the ubiquitously present protein called ubiquitin, so causing ubiquitinated proteins, which are then taken to a complex called proteasome, and the proteasome is going to digest the ubiquitinated protein and release the protein fragments. So that is regulation of eukaryotic gene expression. Hope this was helpful.